Santa Cruz in the Camps Lab of Computational Biology. Today I'll be talking about identifying selections operating on HIV reverse transcriptase via uniform manifold approximation and projection. This work was done in collaboration with Manal Camps and JChem. HIV-1 has evolved resistance to several antiretroviral drugs, and in 2020, AIDS was still causing 680,000 deaths worldwide. The error-prone reverse transcriptase enzyme, which does not have proofreading activity, represents an important source of mutations. Since HIV mutates so rapidly, it would be useful to have a model of which mutations act together to cause drug resistance. Currently, we only have observation-based guesses from specialists about which mutations act together to encode resistance to certain drugs, but it would be good to have a data-driven model that explains this. Several efforts to map the epistasis of HIV's drug resistance mutations have been reported using methods such as multidimensional scaling and principal component analysis. These analyses define a pairwise distance matrix along positions on the HIV genome to create a distance matrix, then perform an eigenvalue decomposition on this matrix to observe how the mutations relate in lower dimensional space. However, eigenvalue decomposition is inherently a linear dimensionality reduction, which limits the identification of large interdependent networks of mutations that are evolving together. Here, the hope of capturing such interdependencies, we turn to a nonlinear dimensionality reduction technique called UMAP, which allows for the identification of relationships between an unbounded number of mutations simultaneously. The 72,000 reverse transcriptase sequences used in this analysis were sourced from the Stanford HIV drug resistance database. Primarily, these sequences mapped the P66 subunit of HIV-1 reverse transcriptase consisting of 560 amino acids. These sequences were taken from patients in longitudinal studies from patients under different treatment regimens. For each sequence, we had the patient ID, study year, and list of the HIV treatment drugs administered to the patient prior to the sequencing. Here, we'll refer to this additional information frequently as the sequence's metadata. The various studies represented sequenced different regions of the reverse transcriptase, which raised the need for multiple sequence alignment. The sequences were translated from nucleotide sequences into amino acids using BioPython, and then were aligned using NAFT. Following some quality control filters, we were left with 14,651 sequences. The data that was then prepared for analysis by splitting into the training data, which consisted of just the raw sequences, and the metadata, which consisted of just the rest of the data fields. We finally one-hot encoded the training data of the sequences to prepare for analysis. The novel nonlinear dimensionality reduction algorithm, UMAP, was utilized to obtain a projection of the training dataset onto the lower dimensional space. In this lower dimensional space, each sequence is represented by a single point. dbScan was chosen as the clustering algorithm for this analysis, as it does not require the specification of a fixed number of points or sequences per cluster. In our analysis, this configuration resulted in 21 distinct clusters, of which 2 were considerably larger than the others. For each cluster, we now had a group of sequences which had been brought together based on some latent factors identified by the algorithms. We performed several tests to assess the biological relevance of the clusters that were obtained. The first one was to measure the phylogenetic relatedness of the clustered sequences. To this end, we computed a UPGMA tree for each cluster. The maximum depth of the tree represented the distance of the furthest removed descendant of the common ancestor, and thus this was used as a metric for comparison between the trees. The results are shown in the table 1 on this slide and in this column. And as expected, we observed that the sequences in our clusters tended to be more phylogenetically related than randomly selected sequences, up to 14-fold. Another way to look at the ability of our analysis to identify related sequences is to look at the penetrance of all of the mutations found in each cluster. To measure the consistency and the representation of mutations within a given cluster, we defined a homogeneity index, comparing the mutations with the frequency greater than 50% to the total number of mutations that were observed. The result is shown in this column, and as expected, we see a substantial negative correlation between the maximal UPGMA tree depth and the homogeneity index. which in other words means that the more related that the sequences are, the more likely a given mutation is to be highly represented in the cluster. We hypothesized that some of the clusters could identify different selections. Here we showed the correspondence between mutation and treatment profiles. Each row represents a UMAT-derived cluster of HIV-1 reverse transcriptase sequences, and shown on the left are the frequency of the mutations by position, and shown on the right are the frequency of prescribed drugs in the treatment regimen for each patient. So interestingly, we observed that each of these 8 clusters that we are showing here identified patients selected by a consistent drug treatment. In an effort to show the effort of drug selection, we looked at cluster 2 sequences because we had a good representation of patients sampled before and after treatment. The results of this analysis are shown in Figure 4, and in this analysis we see a high representation of the main NRTI and NRTI resistance mutations. These results point to the scenario where the selection is being driven by drug selection, but it is the network of interactions with the adaptive mutations rather than the adaptive mutations themselves, which are determining the UMAP-derived signature. In conclusion, we used the UMAP analysis to embed HIV RT mutation data in lower dimensional space. We also visualized the results using dbScan, which produced 21 clusters. 3 lines of evidence actually supported these clusters' biological relevance, including phylogeny, chronology, as well as treatment. This was striking because the treatment information was not used as input into the analysis. And the mutation profiles associated with each cluster likely represents the higher order epistatic context supporting these evolutionary pathways, which is information that isn't generally accessible by other types of mutational codependence analyses. Thank you.